Hello everyone. We have started a video series on the topic cell cycle. In my last video, I discussed in detail about the first phase of the interphase, that is G1 phase or the gap 1 phase or post-mitotic phase or pre-synthesis phase. It is the phase where cell prepare itself for the S phase. Okay, and what type of preparation generally occur in the G1 phase? Transcription process take place. The DNA is converted, or DNA is used for synthesis of the RNA. Then from the synthesized RNA, new proteins are produced by the process of translation with the help of ribosome. Then both these processes require large amount of energy in the form of ATP. So cell also produce energy by the process of phosphorylation. And due to all these things, the size of the cell increased, and finally all the other requirement which is involved in the replication process are collected in the G1 phase. So after successful completion of the G1 phase, now cell come here, and at this point cell check whether it has all the necessary requirements which require for S phase or which require for replication, and if all the material is present at this point, now cell get green signal to enter into the S phase. Okay, and once cell enter in the S phase, the process of DNA replication occur. Okay, we know that every living cell show accurate mechanism by which cell replicates or cell copy its genetic material, and the process called DNA replication. The process occur in three different stages like initiation, elongation, and termination, and different enzymes, different proteins take part in every process like primerases, helicases, topoisomerases, different type of DNA polymerases, DNA ligases. All these enzymes work together to produce exact copy of the genetic material which cell has. Okay, and the process called DNA replication. And the DNA replication mainly occur during the S phase. So first thing that take place inside the cell at the time of S phase is DNA replication. One should ask why cell replicate its DNA, okay, during the cell cycle. Okay, how DNA replication is connected with cell division. What is the use of DNA replication for cell division? Okay, without a proper genetic material duplication or DNA replication, cell cannot undergo cell division. Suppose if this is the animal cell, like human cell have 46 chromosomes, okay, 44 autosome and 2 sex chromosome, or say Drosophila melanogaster have 4 chromosomes, or say domestic cats, okay, cat have 38 chromosomes. So suppose this is the cell of a domestic cat, okay, animal cell, and now this animal cell have nucleus, and inside the nucleus this cell have 38 chromosomes. Okay, out of this 38 chromosome, 36 are autosomes, which is responsible for production of the different morphological characters of the cat, while 2 are sex chromosome, like XX in female and XY in male. So total number of chromosome in cat is 38. Okay, and if suppose this is the cell, and now the cell want to divide by M phase, okay, and after division cell will going to produce 2 daughter cell. Every cell have cytoplasm and nucleus. So suppose this is the parent cell having 38 chromosome, and now the daughter cell also want to receive 38 chromosome each. In that case, first this parent cell have to double its genetic content. Okay, so initially cell have to replicate its chromosome, so that parent cell must have 76 chromosome initially, okay, before entering into the M phase. And now after cell division, each daughter cell will going to receive 38 chromosome in cat. Okay, so without DNA replication, DNA duplication, cell cannot enter into the M phase. And this process of DNA replication should be very accurate. Okay, and this process generally take place in the S phase of the cell cycle. Okay, and all the requirements of the DNA replication, as I said earlier, was collected during the G1 phase. So first important process that occur in the cell during the S phase is DNA replication. After DNA replication, centrioles which is present in the cell also divide, also replicate. One centriole divide into two. Okay, this is the second thing which occur during the S phase. Okay, you should remember that centriole division, okay, occur only in the animal cell, as plant cell do not have centrioles. Okay, in case of animal cell, now centriole suppose present here. At the time of S phase, at the time of DNA replication, centriole also divide and two centrioles are produced. And this is the part from where spindle fiber will going to arise at the time of actual cell division. So centriole will going to play important role in the later half of the cell cycle. Okay, so centriole division occur in the animal cell during the S phase. And the last and important thing which take place during the S phase is histone protein synthesis. What is histone protein? Histone proteins are the positively charged protein, okay, which is present inside the nucleus of the animal or plant cells, and histone proteins generally used for wrapping the DNA. Newly synthesized DNA, as genetic material will going to duplicate during the replication, new copies of DNA will going to produced in this process, and this newly synthesized DNA have to be properly coiled, have to be properly placed inside the nucleus, and for proper coiling of the DNA, cell require positively charged protein, as DNA is negatively charged due to phosphate. So the protein should be positively charged, and these proteins are called histone protein, and they are produced during the S phase. Histone proteins are positively charged because they have amino acids like lysine and arginine in large quantity. Okay, so now this histone protein which is produced during the S phase will going to bind to the newly synthesized DNA inside the nucleus, and due to the wrapping of the DNA around the histone protein, a special type of structures are produced inside the nucleus of the daughter cell, and it is called as nucleosome. Okay, the model which show this type of a structure in detail is beads on string model. I'm going to discuss about beads on string model in my upcoming videos. Okay, so what exactly happened during the S phase? Three important things happened. Okay, once cell enter from G1 phase to S phase, the DNA of the cell going to replicate. Okay, the genetic material of the cell will going to double. Then centriole division occur in the animal cell, and now this centriole will going to produce spindle fiber in the later half of the cell division. Okay, and histone proteins are synthesized, and this histone protein are positively charged protein. They will going to used by the cell for wrapping the DNA and placing the DNA at a proper site inside the nucleus. So these are the important event which occur inside the eukaryotic cell during S phase. In my next video, I am going to discuss about different process which take place inside the cell during G2 phase. Thank you.